So good morning, everyone. Hi, I'm Mark Gillies, and here we are again for another Macular May. It hasn't the time gone quickly since last year, and we're going to have a roundup of the things that we're doing, latest developments in research in macular disease. I'll be talking about the new treatment now for macular atrophy, which is a complement inhibitors, which were recently released in Australia. Associate Professor Ling Zhu from our laboratory will be talking about how we get human maculars and culture them in the laboratory to do research on how to find new ways to treat macular diseases. And Professor Matthew Simunovic at the end will be talking about some research he's done on ways to deliver gene therapy to the macula. So, who's this slide here? That's Ling's presentation. Is he first? All right. So I'd like to first introduce Associate Professor Ling Zhu, who's the leader of our laboratory. Ling did a PhD in the United States and came here about 11 years ago. And now he's the leader of our lab and drives most of the laboratory research we do here. So Ling, thank you.
Hello, everyone. I'm Dr. Ling Zhu from the Macular Research Group. Today, I'm going to show how we study the macula using a new approach we call the macula on a chips. I'll talk about what we have discovered in the lab and what it could mean for patients with macular diseases. So first, we recognize and pay respect to elders and communities past, present, and emerging of the land. Many of you know about macular diseases. You might wonder, why does the macula get so many diseases? The macula is small, only a few millimeters across, but it's the central high-definition zoom of our vision. It's like the spotlight on the stage, handling brightest and the most detailed tasks. Hence, it's prone to wear and tear, making it more vulnerable to damage and disease. To study macular diseases, we need a model as close to human macula as possible. Mice and other laboratory animals don't have true macula at all, so they can only tell us so much. Instead, we use donated human retina tissues in our lab. We keep a small piece of human macula alive in a dish, essentially in a mini macula that is still living and active. This tiny patch of the retina behaves much like it would inside the eyes. It even responds to light. Of course, it lacks of blood circulations and some cells connections. But despite of this limit, it offers an invaluable window to human macular health. Why this is important? Because it lets us watch how a human macula ages and develops diseases in real time. When we can see how its cells change when trouble strikes. So we can test ideas for protecting or preventing them, or in actually human tissues. In short, it is a tiny living testbed for treatment accomplishing the animals and the models cannot. So keeping a piece of human retina alive outside the body is challenging. Our solution is what we call the macula on a chip, also called MOC systems. It is essentially a mini life support for the macula. We place a donated macular tissue in a special chamber with nutrition and oxygen, carefully controlled the environment in the incubator. This setup keeps the macular tissue healthy for over 5 days. In those crucial days, we can perform experiments on it, such as adding a nutrition or chemical, and observe how the macula responds. In short, the macula on a chips allow us to do human macular research, something that wasn't possible with traditional animal models. One of the diseases we deeply focus on is called macular tail agitation type 2, or MACTAIL. It is a rare condition that affects the macula, interfering with everyday sight. To tackle such a complex condition, I have had the privilege with working with 4 dedicated research scientists from our group, Dr. Ting Zhang, Dr. Rita Zheng, Dr. Qing Chen, and Dr. Shaoxue Zheng. Each bring their own expertise in retinal metabolism, molecular biology, and ophthalmology. And together, we use our macular-on-a-chip systems to study this disease at a deeper level than ever before. Let me give you some quick background on what we already know about the MacTail. MacTail really happened in an oval shape, surrounding the center of the retina, a zone known as the MacTail zone. And one of the earliest signs of the trouble in this area is loss of Mueller cells, the essential support cell of the retina. These Mueller cells are like scaffolding and caretakers. They keep the retina environment stable and recycle waste and support the health of the neighboring cells. In the eyes of the MacTail, we see few of them. So studies have shown that loss of Mueller cells makes the macula and the photoreceptor appear to the bare and no longer protected by the ural support. This loss sets the stage of the more damage follows. But what caused this Mueller cell disappear in the first place? That's a question we set out to answer. MacTail affect eyes built up a neurotoxic fat molecules called deoxysphingolipid. These are toxic waste products that accumulate in the retina. They may arise from metabolic imbalance. The retina usually use both amino acid, serine, and alanine to build its fat and proteins. But the MacTail patient have abnormal low serine levels in the cells. Instead of using, so with too little serine, the cells use alanine instead, and it end up create toxic byproduct called deoxysphingolipid. Essentially, the cell run out of their main ingredient, such as serine, and substitute it with alanine, creating a poisonous byproduct. Over time, these toxic molecules build up and likely begin harming the cells. To understand why macula is so vulnerable to MacTail, we use our macula-on-a-chip systems and conduct a series of experiments using real human retina tissues. In this experiment, we study how the macula process glucose compared to the peripheral retina. We use isotope-labeled glucose to trace how the downstream metabolites happened in both macular region and peripheral regions. What we found was striking. The macula produce more alanine, and the alanine to serine ratio is the serine to alanine ratio is much lower than the peripheral retina. This matters because that imbalance, less serine and more alanine, makes the macula more prone to produce toxic deoxysphingolipid. So in our next experiment, we directly measure those lipids again. Using the Mark systems, we profile the retinal lipid profiles, compositions, and discover what the macula does. In fact, and discuss the macula actually does in fact accumulate significantly more deoxysphingolipid than the peripheral regions. We can see all these red bars means the deoxysphingolipid detected in the macular region, and the green bars is detected in the peripheral retina. In other words, the chemical imbalance we saw in the first experiment may be driving this toxic buildup. These two experiments together reveal a critical insight. The macula in nature sits near a metabolic tipping point. Even under normal condition, it's poised to overproduce toxin if serine level drops. And that may explain why disease like MacTail strikes the macula first. It's a part of the retina most vulnerable to this toxic shift. To confirm these toxic molecules, deoxysphingolipid, will indeed cause harm, we exposed the health-donated macular tissues to deoxysphingolipid in our Mark systems, trying to recreating the chemical conditions of MacTail in a dish. What we saw was striking. The toxic lipids damage the tissue, and they especially harm the Mueller cells. We can see all these green dots suggesting the dying cells, and these dying cells co-localize with the Mueller cell marker, suggesting the Mueller cells in the retina is dying. So this was crucial evidence that the deoxysphingolipid are the cause of actively injuring the retina cells and driving the disease process. Once we discovered the toxic lipids like the deoxysphingolipid were harming the retina, especially the Mueller cells, our next question was, where exactly these molecules being produced and what is the source? To answer these questions, we zoomed in the key enzyme responsible for making alanine, which we called is ALT, short for alanine transaminase. The immunostaining found that the expression of the two isoforms of the ALT are mainly in Mueller cells. Armed with this insight, we asked, can we interrupt this cycle? Can we stop Mueller cells from overproducing alanine? That led us to our next experiment, where we use a compound, which is the beta-chloro-L-alanine, or BCLA, to block the production of alanine by inhibiting the ALT enzyme. When we added BCLA to the macular-on-chip system, the result was encouraging. First, we observed the alanine level significantly dropped, and then the serine to alanine ratio improved, and the retina, especially Mueller cells, appear healthier and more stable. In other words, by rebalancing the retina's chemistry, we could reduce the production of toxic sphingolipids and help protect the cells most at risk. This approach offers a promising path forward. If we can fine-tune the retina's metabolism, whether through drug or supplements or another intervention, we may one day slow down or even prevent vision loss in conditions like McTell. So what our research shows so far? The macula is has unique metabolisms, make it more likely to build toxic fat deoxysphingolipid. The toxic fat seems to damage Mueller cells, which is the key supporters of the health vision. Our lab model, macula on chips, help us test how these changes affect real human macula. We may be able to protect the retina and slow down disease by adjusting certain metabolic pathways. All these discoveries are the result of the team effort. I'm lucky to get the support from our director, Professor Mark Gillies, and our collaborators, Dr. Meidong Zhu, Jianhai Du, and Marin Gardner. So most of all, none of this would be possible without generous donor who donates their eyes tissue to research. We are deeply grateful for those donors and their families. By combining human tissues research with the innovative ideas such as macula on chips, we are charting new ground in understanding and protecting the gift of the sight. Thank you.
Thanks very much, Ling. We'll hold questions until the end. So now I think I'll go on to the next presentation, which is on macular atrophy. Yes, on macular atrophy. MacTel is a type of macular degeneration, but it's not age-related macular degeneration, which is the classic form, which is so common. So today I'm talking about macular atrophy. If we just have a look at what that looks like first. So this is the way we image the macula in the clinic. There's various imaging modalities. Oh, that's not it. Modalities. Hold on. Sorry. This one. There's various imaging modalities here, but basically we're looking through the pupil at the macula. And if you top left here is a color photograph, which we look when we see in. And if you look at the edges, that's normal in all these images. But in the center, you see all this. In this case, you see this sort of white cloud. It's called geographic atrophy because it looks like the map of some country that you've never seen. So that's what it looks like when you look straight at it. When you take a cross section from it, you can see here the retina is divided into the inner retina and the outer retina. The light's coming from the top, and the photoreceptors, for some reason, are at the bottom. So that's the outer retina. So this dark band here has got the photoreceptors and the pigmented epithelium that sits on it and nourishes it. Now, in geographic atrophy, the inner retina is still there, so all the relay apparatus is still there, but the photoreceptors have gone. You can see the light is shining, the signal is shining through into the base of the eye here because the retinal pigment epithelium is no longer stopping it penetrating into the deep layers there. So where you see this penetration, these are holes in the retina that don't see. So this is color imaging, this is autofluorescence imaging, another way to look at it, angiography. This eye here has still got the central vision there, that little bit in the center. So this eye may be seeing normally on a chart, but it wouldn't have good visual field on either side. So they'd have difficulty with reading and many other tasks as well, even though they've got normal vision on a chart. So if we look at this in terms of the significance of geographic atrophy, it is one of the leading causes of blindness in the world. It affects about 5 million people worldwide, and it accounts for about 20% of loss of vision from age-related macular degeneration, about 100,000 people in Australia. And it refers to the loss of the photoreceptors and the retinal pigment epithelium in eyes of elderly people with age-related macular degeneration. So if we think about age-related macular degeneration, it starts out as early on the left there. We just see three little spots there. Intermediate, there's more debris under the retina. But these two forms don't affect visual acuity all that much. And they generally, you still have driving vision here. But then from intermediate, you can progress to advanced, which is either atrophic or wet. We also call atrophic advanced dry degeneration. So this happens in about maybe 1 in 10 eyes a year who've got intermediate macular degeneration. The bleeding, of course, we can treat very well now with the injections, but up until now, there hasn't been any significant treatment for the for atrophic macular degeneration. So what causes atrophy? It's a complex interplay of genetics, and the genetic abnormalities is probably about 70% genetic. The genetic abnormalities are mainly in this system called the complement system, which we'll be talking about in a moment. In terms of physiology, the greatest risk factor, of course, is age. The older you are, the more likely you are to get it. And people who are being treated for wet degeneration also eventually get dry degeneration. The environmental causes probably only account for about 30% of cases, and the main one is smoking. There are some dietary influences as well. So typically atrophy, as I said, it doesn't affect the visual acuity to begin with. It often starts to the side of the centre and spreads that yellow part there and slowly goes around the centre and spreads into the centre over time, giving progressive loss of vision. But as I said before, even patients with normal vision on the chart may have impairment of all sorts of other visual functions because the atrophy is all around the centre, but not yet quite at the centre itself. When we look at people in the clinic, we can tell in many cases there are indications of how, this is going to grow. And one of these is the autofluorescence. This is a type of imaging that comes from this retinal pigment epithelium. If you shine on it, it fluoresces and you pick up the signal. So if it's black, that means there's no retinal pigment epithelium, or RPE is no longer there. So that's what atrophy looks like. But this atrophy, there's no whiteness around there. When you look at on the right here, you can see all these white areas. So on the autofluorescence, cells tend to go white before they die, then they go black. So if you've got a lesion like this with no white edges, that's unlikely to grow. So you probably wouldn't want to treat that. But if you have a lesion like these on the right here, who've got these white edges, and this one here, these are the ones that can grow quite quickly, and these are the ones that you're more likely to want to treat. The other problem with atrophy is that it's generally a bilateral disease, so you get it in both eyes, not just one eye. And there's the average time to development of atrophy in the second eye is about 7 years after developed in the first eye. So these are the sorts of things that we're up against with people with atrophy. One of the major problems they have, even though they don't look blind because they're still looking around using their side vision, they can't recognize faces. They can't do many of the daily activities that they need to do, such as reading and driving, so they lose their independence. This has effects on their mental health. They avoid going out or unfamiliar places, and they're worried about their disease. So there's all these things, all these challenges confronting people with atrophy. At the moment, there's very little that we can do apart from telling people they should stop smoking. That has a very strong effect. You can change your diet, but it's probably what you've eaten for the last 50 years rather than what you start eating now. But leafy greens, obviously, what you look. People who are fit and well and not overweight generally have fewer illnesses, so exercise and that sort of thing is helpful. the age-related eye disease supplements like Macuvision or those sorts of things, they can slow it down a little bit. And low vision aids actually something where there's a lot of development these days with the technology, with low reading glasses and these sorts of things. And there are going to be quite a few developments there, but they don't fix what's going on in the actual macula. So now, what would we like to do? So there's talk at the moment now about this Muller cell reprogramming. So you just heard about Muller cells from Ling. There's two types of cells in the retina. There's neural cells, and there's which pick up the light, and there's Muller cells which support the the neural cells. So there's a Muller cell in green there, that big one. Now, it's possible to turn them into photoreceptors, and that's what happens in reptiles and lower order species, where if you injure one of these cells, the Muller cell will start turning into a photoreceptor. But this is blocked in humans. There's a couple of genes which regulate this process, which are strongly expressed in humans, and no one's worked out how to turn them off yet. But one of these genes is PROX1, which is recently identified by a Korean group, which you may have heard in the paper. And in mice, they've had some experiments in models of atrophy where they've given them a PRX1 neutralizing antibody, and they've been able to regrow retina. So that's where we are at the moment with giving you your vision back again. And they're hoping to get into trials in 2028. The problem is that a lot of things work in mice that don't work in humans, but that's where the most advanced that is at the moment. In the meantime, all we can do really is try to slow down the expansion of these lesions, and that's where these complement inhibitors come in. So this is a diagram. Complement pathway is a very complicated pathway which most people don't understand except for super specialists in the field. But basically, it is a part of the immune system that regulates local immune defense and becomes dysregulated in eyes in people with AMD. They often have genetic defects in genes associated with the complement pathway. So here's a complement pathway here, and you can see a complex series of interactions which comes down here. And when it's overregulated or overactive, as it is in AMD, you get inflammation, cell death, and the cells all sort of get eaten up. And the pegcetacoplan, the new molecule, or Syfovre, or what it's called, is a C3 blocker, and it blocks the activation of this pathway. They've had many complement activators, inhibitors, gone through clinical trials that didn't work, but this is the first one that actually worked. And it was approved by the FDA about 2 years ago in the States and was not approved in another country around the world, except apart from the TGA, who approved it about a month ago. So now complement is not reimbursed yet. The government's not paying for it, but you can get it in Australia. So let's look at the clinical trial data on complement. And there are many, many graphs, but this one pretty much summarizes what the effect is. So if you have a look at this line here, the gray line is the people who got sham, and this is the expansion of their areas of atrophy over a 3-year period. And in the pegcetacoplan or Syfovre arm, there were 2 dosing regimens. One was every month and one was every other month. And every month tended to slow it the best in orange here, but every other month, every second month was also effective. So in the whole study, it was about a 1/3 reduction in the rate of expansion of these atrophic lesions. But interestingly, the rate of expansion went up after the second year. So maybe the first couple of years, the cells that were all going to die, they've all died. And now maybe you're protecting cells after 2 years, which means you'll get a greater effect. In any event, this is the 3-year data of pegcetacoplan, which is what all the fuss has been about. This video here gives you an idea of what the drug can do. So on the left, on your left is a treated eye in the study, and on your right is the fellow eye. And this is the expansion of the lesions over 1 year in both eyes. So you can see the lesion, and the one on the left is expanding slowly, and the one on the right is much more rapid expansion. Presumably that is the treatment effect. I've got another one here, where if you have a look at the one on the left here, what we're interested in is the center, because the center is where the central vision is. In the one on the right, you'll see the center gets involved very quickly, but on the one on the left, you see the center is preserved all the way out to the end of this study anyway, which means the patient lost the vision in that eye you're seeing on the right, but they maintain their central vision in the eye on the left. So what about the visual outcomes? The problem is, and the reason that they have such trouble in approving it, is it did not improve visual function. So this is the as measured on a visual function chart. As I said, you can lose a lot of macula through atrophy and still have normal visual function. So it's not a great measure. And they didn't find anything here. But atrophy is like glaucoma. You don't actually improve someone's vision by giving them glaucoma drops. You prevent the loss of vision, which we know will come if you don't control the pressure. And this is the same thing here. We give the injections to prevent the loss of vision, which we know will come if you don't stop the expansion of these atrophic lesions. And 2 years wasn't long enough for this study. If, however, you looked at the eyes where it hadn't involved the center yet, and it was quite close to the center, but it hadn't involved the center yet, in the orange here, there was a significant improvement in those patients' eyes compared to the people that did not get treatment. So just going through now, that's the benefits of the treatment. What about the safety? With all these new drugs, we worry about inflammation. There are some safety. Well, the first thing actually is wet macular degeneration. In eyes that don't have wet macular degeneration, there was an increased risk of getting it during the treatment over the 2 years of the study. So the sham-treated eyes who didn't get it, only 3% of them got wet macular degeneration during the 2 years, whereas the ones who got it monthly, it was 12%. These eyes continued to get treatment with normal VEGF inhibitors, and they did reasonably well. But that is one risk of treatment. There's about a 10% chance that you'll induce bleeding from the wet degeneration, which you can get anyway. The second thing is intraocular inflammation. All the new drugs coming out now for macular degeneration have an increased risk of intraocular inflammation from the drug, which is more than we saw from the drugs before. We don't quite know why. From Syfovre, there is one very severe inflammatory event which can destroy all the vision, and it occurs on the first injection, and it occurs in 1 in 4,000 eyes. So generally they'll give it to the worse eye first, and if they don't get that reaction, they'll proceed with treating the better eye, which is the aim of the treatment. They didn't find any evidence of non-occlusive retinal vasculitis, but they in the studies, but they have been reported, and this could occur in about 1 in 10,000 people. It's a rare but severe complication. So there's a bit of inflammation associated with these new drugs. However, it is the first drug that we've had that will actually slow down the rate of expansion of these lesions of atrophy. I think some patients, if they are very well informed, know exactly what they're getting themselves into, may benefit from this intervention. But basically, they're probably the ones who've lost vision in one eye and they've got atrophy, which is fairly close and threatening to spread into the centre of vision of their second eye. So just to conclude that, we've discussed that geographic atrophy for age-related macular degeneration is a major cause of loss of vision worldwide, which is also a major unmet medical need. The cell-based therapies, which are the ideal cure for this, are being worked on and actively researched, but they're probably at least 5 years away from the clinic, if that. This new drug, pegcetacoplan, Syfovre, is the first drug that's been released, which will actually slow the rate of expansion of the these atrophic lesions. It has to be given approximately every 6 weekly indefinitely to realise the benefit, but some patients are very likely to benefit from this. But the observational studies, such as the Fight Retina Blindness study, which is what we've built modules to track this now, we'll be watching the outcomes of this very closely. So thanks for your attention on that. And just for the last presentation and then questions after that, Professor Matthew Simunovic.
Thanks very much, Mark. I'd like to thank Cami, who's standing up the front here for arranging everything. I'll be talking about approaches to delivering subretinal gene therapy once I get my slides up. There we go. So I'll be talking about a trial which we conducted here at Sydney Eye Hospital and the University of Sydney called 1-2-SIT, which compares two different approaches to delivering therapies underneath the retina. And most of these therapies are directed at the macula in particular. And so it's called the 1-2 sub-retinal injection trial. Click on it. Yeah. Okay. Sorry about that. So what is subretinal injection? It's an approach to delivering therapies which is mandated by certain emerging therapeutics, in particular retinal cell therapies like gene therapy, stem cell therapies, as well as retinal gene therapy. And we perform this procedure after we perform a pars plana vitrectomy, which is a micro keyhole operation where we remove the gel that fills the inside of the eye using small instruments, which are the size of small needles. And in the early days of developing this as an approach, investigators standardized the steps for performing the procedure in the hope that this would mean that a fixed amount of therapeutic solution would end up underneath the retina. But since the early days, variations have emerged amongst surgeons in how we perform these procedures. And in particular, there are two variants which are used, the so-called one-step injection, in which the therapeutic solution itself both sort of defines the region where we need to deliver the therapy and delivers the therapy itself, and a two-step procedure which was developed by one of my colleagues at Oxford University, Robert McLaren, whom I worked with over a decade ago. And in the first step of a two-step procedure, we first of all inject an inert solution. This is shown in the bottom right here in a surgical video of something called balanced salt solution. And we're able to use a technique called optical coherence tomography, which Mark spoke about earlier, on the table whilst we're operating to confirm that we're in the correct anatomical location. This is important because we want the treatment only to reach this space. The correct anatomical location is a potential space. It's not normally there, and it's sandwiched between the nerve layers of the retina and the supporting layer, which Mark also was talking about, the retinal pigment epithelium. Until recently, we literally had no idea about how much therapeutic solution when we're performing this procedure is wasted, that is refluxed back within the vitreous, the internal compartment of the eye. And so there's a lot of conjecture that still remained about which might be the best approach. This is important because these therapies are expensive. So the only approved therapy which is delivered via subretinal injection is Luxturna gene therapy. It's the most expensive medication, milliliter per milliliter. Each eye costs $500,000 to treat, and that's for 0.3 of a mil of solution. So literally until recently, we had no idea about how much drug is refluxed when we're performing subretinal injection. And the first data we have came from a study which was conducted in North Carolina by Sue and colleagues, and they used a model of a pig eye, a cadaveric pig eye, and they found that reflux estimated via, again, OCT was significant, somewhere between 12% at best and up to 96%. So that's a lot that's being wasted. And more recently, colleagues performed a similar study, except this time instead of doing a one-step injection, they used a two-step injection in primates of gene therapy. And what they did is they counted the number of viral particles which were delivering the gene therapy in the internal cavity of the eye after the injection. And they found that reflux was lower at somewhere between 0.08% and 1.25%. And we've developed in Sydney an approach which uses something called sodium fluorescein, which is a fluorescent salt, which we use in ophthalmology all of the time, both on the surface of the eye and in performing ocular angiograms to label drugs that we inject underneath the retina. And I've got an example of a subretinal bleb where I've injected a clot-busting medication called TPA, or tissue plasminogen activator, which has been optically labeled with sodium fluorescein. So you can see qualitatively exactly where the medication goes. It's a bit like those of you who remember Mrs. Marsh and her chalk and plaque. It's similar to that. So we can visualize a normally transparent medication going underneath the macula in this case. And we've taken it a step further in the lab by using this approach to quantify how much drug is refluxed back within the internal cavity of the eye by performing something called fluorophotometry, which I'll explain in the next slide. So we collect at the end of surgery all of the fluid which has been tinted by this sodium fluorescein. We collect it and we send it to the lab. Because we've been injecting it underneath the retina of patients who have submacular hemorrhage, we spin it down to remove any red blood cells. We're able to measure its volume. We use a technique called fluorophotometry, which measures how coloured the solution is. And this tells us how concentrated the sodium fluorescein is. And from this, we can calculate how much the drug has been wasted or been refluxed when we're performing these injections. So we've been using this technique in the trial, which I mentioned earlier on, 1-2-SIT, which was performed here at Sydney Eye Hospital and the University of Sydney. We included 12 patients on the trial. So this is a relatively rare indication that we're using this technique in. And it was the exemplar of patients presenting with massive or large submacular hemorrhages secondary to age-related macular degeneration. And we selected this sample size based upon calculations which were informed by preclinical data and in particular by the study of Zhu and colleagues. And all of these patients had reduced vision. It had to be 2200 or 660, equivalent to legal blindness. They had to have a duration of symptoms of 7 days or less, and they had to have a significant size of hemorrhage. So it needed to be greater than about 1.5 millimeters in diameter in total. And we injected sodium fluorescein mixed with TPA, the clot-busting medication which I referred to earlier, underneath the macula, either as a one-step procedure, which is shown at the top here, or as a two-step procedure, which is shown at the bottom here. At the end of surgery, we collected all the fluid, sent it off to the lab, as I detailed earlier on. In doing this procedure, what we want to do is we want to break down this blood clot and we want to displace it away from the macula to the edge of the retina, where it's going to have a less detrimental effect on vision. And to help facilitate this, we inject a gas bubble into the eye. We also want to switch off the process of bleeding, and so we therefore injected an anti-VEGF agent to do this at the end of surgery. We followed patients up for 3 months following the procedure, and patients underwent monthly anti-VEGF injections. What we were really interested in this study was the proportion of drug that's refluxed when performing these two different approaches to subretinal injection, but we also looked at other outcome measures, in particular how long it took overall to perform the surgery, what the visual outcomes were like, and what the structure of the macula was like. And we published the protocol in advance in British Medical Journal Open. And in terms of the primary outcome measure, this is summarized here. That's for the one-step procedure. This is for the two-step procedure. So on average, there's no difference in reflux between these two procedures, 4.8% for the one-step and 3.9% for the two-step procedure. However, if you look at the variance, the scatter of the data, it's greater for the one-step procedure than it is for the two-step procedure. And this really matches with surgeons' experience. So the most difficult part of performing these injections is the initiation of the bleb, getting the fluid underneath the retina and opening up that potential space, which I referred to earlier on. And if you're using the therapeutic solution, this is going to be an issue because you're wasting that therapeutic solution. If you're performing a two-step injection, you're raising the bleb initially with balanced salt solution, which you're infusing into the eye in any event, and it doesn't matter how much of this you waste. In terms of all of the secondary outcome measures, there's no statistically significant difference in the overall duration of surgery. It was slightly longer for the two-step procedure at half an hour versus 26.8 minutes for the one-step. There were no differences in either functional or structural outcomes in the study. We're also interested in whether or not this technique could be applied to gene therapy and cell therapy, both in clinical trials and clinically in the future. So explored whether or not adding sodium fluorescein affected viral counts for retinal gene therapy. So that is the number of virus particles in solution, which would be effective, as well as cell viability for cell therapy. And the take-home message is that it doesn't appear to affect either. And so this implies that this technique, which we developed in our lab, might be used in the future for both retinal cell and gene therapies. So in summary, we've seen that we can estimate in humans in vivo in life drug reflux following subretinal injection using sodium fluorescein as an optical label. We've seen that it's suitable for assessing drug reflux when performing subretinal injection for either cell or gene therapies. We find no statistically significant differences in average reflux between the one-step and the two-step procedures, nor are there significant differences in duration of surgery or anatomical or functional outcomes. However, two-step subretinal injection appears to be associated with significantly less variability than one-step injections, and for this reason, we believe it to be the preferred technique. The study was published online this month in ahead of publication in paper format in American Journal of Ophthalmology in June. I'd like to thank my collaborators, in particular Dr. Lei Kuhn Tu from my lab, who's at the back and here today, as well as my collaborators at Sydney Eye Hospital and the Foundation Fighting Blindness, who supported the work. Thank you.
Thanks, Matt. So that's three different topics. Does anyone have any questions about what we've been presented this morning? Yes, ma'am.
Mark, those donated tissues that you receive, do they deteriorate over time?
In the if you cut out, when we get them, generally the person died, maybe about 12 hours ago, and they're starting to fall apart. Then the retinas you look under a microscope. But if you put them in this perfused medium, so the medium's continuously flowing through with all the right ingredients, then it comes back to life. And we're not the only people who've established this, but there's not that many people around the world who are doing it. But there's a Hungarian group who'll keep them for 3 months. But they will go on for quite a long time. But we usually do the experiments within the first few days because they probably change after a couple of months. Yes.
The treatment that you talked about that just got approved by the TGA, how much is that costing patients per injection?
Good question. Syfovre is very expensive. If you import it from at the moment, it's directly imported from America at American prices, which is about $3,200 per shot for the drug, without the surgery, without the injecting fees and everything. Now, I know that Apellis is seeking for reimbursement, and they probably won't get blanket reimbursement for everyone with atrophy, because if the atrophy is a long way from the centre and it's going to take 5 years to get there, then you probably don't want to have 6 weekly injections to slow that down. And if you've got normal vision in the other eye, so even if you save that eye, you still haven't done the patient a whole lot of good because they've still got the vision in the other eye and they may still be able to drive. So they'll focus it down. It will probably have something to do with an impairment in one eye, and the atrophy is threatening to grow into and would likely get there within 3 years if you can't slow it down. It'll be something like that. And then the TGA will just have to, they've got a certain amount of money and drugs from all over medicine that they're more and more expensive these days. So we'll just have to see what their judgment is. Yes.
What's been the uptake with this drug since its approval about a month ago? Has it been well received by patients?
I don't know what it is in Australia, but certainly there's a lot of Syfovre was given in America, and a second one has come out in America now called Izervay, which has less data, possibly a little bit less inflammation associated with it, but still some inflammation, and that's still being people are still that's still being dispensed quite on a widespread basis. What I heard was a lot of people have started it, and then there's not so many new starts now. So a lot of doctors started some people when they first came out, and a lot of the medications are going on the people who first started with it. And they're getting experience with it. It's a long, slow process. It's a benefit that becomes obvious over years.
Adherence to treatment been pretty good? Because I feel like 6 weekly injections is quite a lot for.
The adherence to treatment is not going to be incredible. So with VEGF inhibitors, if you've got a wet macular degeneration, generally if you give the injection, the vision actually gets better. And then when the injection wears off, the vision starts going off again, you get another injection and it gets better again. So that's highly motivating. That doesn't occur to anyone with atrophy. In atrophy, the vision stays the same. It never gets better. It just deteriorates more slowly. So you have to have a very well-informed patient. You want to have a lesion which you've already had some scans on it from a year or two, and you can confirm that it's actually growing, which is important. And, it's not too far away that it's not even worth treating because it's not going to get there for years. So there's a lot of things to take into account. But some patients it will be beneficial for, but many patients it probably won't. Oh, these are the pre-questions. Yeah. So do any of these have medical updates? Diabetic retinopathy, diabetic macular edema, detached retina. Right. Okay, that's a big one. I'll think about that. Do you want to take the second question?
Sure. So how can those with macular dystrophy improve their vision? Look, the tried and tested ways which Mark has sort of touched upon already. So optimizing things like their refraction, low vision aids. There are new medical therapies which are being developed, as well as emerging treatments. Mark referenced and I referenced cell therapy. There are also next generation electronic retinal implants. So one in particular called the primary implant was approved in Europe late last year. It was developed by a group in France called Pixium Vision. They're looking at implanting the device into some patients in Australia in the not too distant future as part of this special access scheme via the TGA. Want to go back to question one?
Yes. So any medical updates for diabetic retinopathy? Diabetic retinopathy is pretty much hand in hand with wet macular degeneration, because we also treat that with VEGF inhibitors. So that is that's all about the newer agents with longer duration of action, the Eylea 8 milligrams and the Vabysmo, which has blocks another thing. It also blocks Ang2. These are the second generation VEGF inhibitors, and they're also being used for diabetic retinopathy, diabetic macular edema. There are a couple of new targets for diabetic retinopathy, but they're still in clinical trials, so it's probably not worth saying too much about them here. There's another question here about the failure of the Opthea trial. So the Opthea trial, that was an Australian initiative originally. VEGF has these different isoforms, A, B, C, and D. And the VEGF inhibitors we use mainly block VEGF-A. Opthea was an Australian drug that blocked C and D. It was promising all the way up into phase 2 studies, but it failed the phase 3. And the patient who asked the question, who's a mathematician, was disappointed by that and couldn't work out how it could pass a phase 2 and not a phase 3. Well, it happens. In about 50% of cases, eyes that go to the phase 2 go to the phase 3. The reason is there are two different types of study. There's more leeway for optimism from the investigator in a phase 2 study because that's not what actually triggers the registration. So they're not quite as strict with those. But with phase 3 studies, they're very strict, and they're more limited in the ways they can analyze the data in a way that gives them the result they're looking for. And they were unable to find the result they were looking for. It was a phase 3 study, and that's just that. As I say, about half the study, the drugs that come to phase 2, that pass phase 2, fail to phase 3. And in fact, in atrophy, that's been particularly the case with several complement inhibitors passing the phase 2 but failing in the phase 3. Syfovre and Izervay are the first complement inhibitors to actually pass this process, but there've been clinical trials for years with complement inhibitors. This is not the first one. I think that's about. Any other questions? Yes, Michelle.
Yeah, thank you. Thanks for the great talks. They're really interesting. Along the same lines of updates, I wondered, is there anything new coming for drug delivery? We've got injections and so on, but I wondered if there was anything new coming up clinically for.
Well, the big thing that we're interested in is vesicles to deliver mRNA interventions, which are the COVID vaccine was an mRNA intervention, and mRNA is an effective way to manipulate genes in the retina. And at the recent ARVO conference, which is a big world vision conference that we all go to every year, there's been a lot of talk about extracellular vesicles, which are these vesicles secreted by cells. They communicate with other cells, and the penetration of these vesicles is excellent and you can sort of target them to the cells that you want to. So we're actually moving into that area of research, trying to design EVs for drug delivery to the retina. Yes.
They're also susvima and the like.
You want to talk about that?
Yeah. So there also, Michelle, there's something that's available currently in North America. We're doing a clinical trial here looking at a sustained delivery system. It's called a port delivery system. It's like a little pot, if you will, that sits within the eye. It's sort of tethered to the sclera, the white of the eye, and it's covered by the clear membranes that cover the white of the eye, the tendons, capsule and conjunctiva. The aim is of this particular trial that we're working on here, Mark and myself, is to refill the implant only every 9 months, ideally.
Oh, that's good. Thank you.
I think that looks about it for this year. There is refreshments up the back. There's some sandwiches there, and is there tea and coffee and stuff?
Just juice.
Pardon?
Just juice and water.
There's juice and water as well. Okay, so see you all next year and hang around for the refreshments.